I can do that one. Thanks very much. It's really great to be here tonight. I read the lineup of the speakers that were coming tonight, and I just thought, shit, I really wish I had IBS right now. Because IBS is funny. Everybody laughs at IBS, and I'm like, nobody's going to laugh at de-extinction science. But the good news is I've been feeling pretty nervous about tonight. So I've had a few queasy feelings, and I could potentially shit my pants at any point. So, I'll definitely keep you posted as we go through. But my lab does work on the de-extinction of the Tasmanian tiger, and it is really about trying to bring back animals that have gone extinct in the past to try and build a better future for our troubled planet. And like I said, it's a barrel of laughs, de-extinction science, right? Like, just read that. How funny is that stuff? That's just comedy from the very beginning. Australia has the worst rate of mammal extinctions of any country in the world, which is absolutely horrific, given what a wealthy country that we are, and that we have a really good state of knowledge of how important all our animals are. But we do have the highest rate. And at that rate, we're predicted to have another 1 to 2 extinctions occurring every decade moving forward. And that's not taking into account adverse weather events. So when we have like a massive bushfire or a massive flood, which is something we're having even more frequently in Australia because of global warming, that is really massively increasing the rate that all these animals are going to go extinct. So we have got to do something about this. Our traditional methods of conservation are not effective enough on their own, and we have to start thinking outside the box at how we're really going to protect the Australian environment moving forward and our incredible biodiversity that we have in this country. So, to do that, I am proposing to bring back this guy, the Tasmanian tiger, which should be walking around on there right now. No, not that guy. This guy. I don't know why it's not playing. Hang on. Okay, so there's a Tasmanian tiger. Just imagine it. It worked in the practice. What is happening? Anyway, he's walking there. He is. I mean, he's extinct, right? Exactly. I saw somebody shout that out. He is, in fact, dead, and that is why he's not there anymore. But these were absolutely incredible animals. I can go back to the first slide, and you can see a picture of one there. So there it is. That's the animal there. Incredible marsupial. So it had a pouch, even though it looks just like a wolf. So really, truly very, very unusual among the marsupial world. But the thing that made it so special was that it was an apex predator. So these are those animals that sit right at the top of the food chain, eat everything else. And what's really unusual about Australia is this was the only apex predator mammal that we had. We don't have any others. So if you think about America and Asia and Europe, they have tons of apex predators. They've got wolves, they've got bears, they've got all the big cats, they've got all the things, right, that kill everything else. We didn't have anything but this animal. So when we hunted this animal to extinction, we wiped out a single only key player in our ecosystem, that animal that's so important in balancing out all the other animals in that particular ecosystem. So massively tragic. So we hunted that animal to extinction in 1936. This was shortly after European settlers had settled in Tasmania. They were farming sheep. They thought the Tassie tiger, if you could see him there, you would agree, looked very much like one of the wolves that they were used to from Europe. And they're like, we're going to kill this guy because he's eating all of our sheep. They were losing a lot of sheep at the time, and so they put about to have a very aggressive extermination scheme. They were paying people to go out and become thylacine bounty hunters, and in the space of about 20 years, they had removed all thylacines from Tasmania. And the black box there is the last one that ever lived, living in the zoo in Tasmania right before it died. So you're just going to have to take my word on that. And, yeah, persecuted for the sheep, but it turns out that people were stealing sheep, so probably that guy's fault. I'm not sure, actually, if he was involved, but he kind of looks Tasmanian. There's something about him. But, they were nicking other people's sheep, and that was really the reason that the sheep were going missing. And so the thylacine, even worse, was persecuted for something that it never did. But when you lose an apex predator, you start to see a destabilisation of the ecosystem. So this other really funny slide that I have about, again, great humour, is what's happening to our Tasmanian devil population. So those animals would have gone extinct in Tasmania if we hadn't intervened as scientists, removed animals from Tasmania, brought them back to the mainland and raised them here. And the reason they're all dying in Tasmania is because they get this terrible facial tumour disease that you can see in that middle picture there. And it's this terrible transmissible tumour that grows on their faces. Unfortunately, these little guys like to bite each other's faces when they're feeding. They also do it socially. When they bite each other's faces, they spread this tumour. Cells actually transfer from one animal to another. It starts a tumour on their face. But what happens is, at the moment in Tasmania, there's no apex predator. So if you're a sick animal like this, there's no one to eat you. So you live in that population until that tumour becomes so big that you literally starve to death or you bleed to death because of the size of that tumour. And that entire time you persist in that population, you're spreading that disease to everybody you come into contact with. When you've got the Tassie tiger there, it eats those guys. They're the ones who are a bit slower, a bit, can't run away as quickly when the thylacine comes in. It eats these guys up, it removes them from the population, and you stop the spread of disease. That's what apex predators do. So this is just one example of what the thylacine would be doing if it was still there. It would stop the spread of that disease. It would have saved this poor little guy from now having to be raised on the mainland, and they translocate them back to Tassie, but unfortunately that tumour just keeps on spreading. So all the animals there are basically doomed as a result of this particular disease. So it's really, really horrific. So if we ever want to bring that incredible ecosystem in Tasmania back into balance, if we want to re-wild Tassie devils again, we have to really think about ways that what we can do in that environment. And the best way that you could balance that back out would be to bring the thylacine back. So we need to start thinking about ways that we can restore that balance. So usually about this point, when I talk about the Tassie tiger, I have a few questions that people always bring up. The first thing people always tell me is they are still out there. So I've had some great experiences in my life. These top two pictures was when I was asked to go on live television. Somebody had definitely found a thylacine. They were going to release the footage live. I had to review the footage and then add the expert opinion to that. The footage was this up here on the top left, which, is clearly the arse of a wallaby. I just unequivocally, and then this guy's going, but look, it's got stripes on it, but that's the shadow of the grass. It's just for fuck's sake, seriously. So there was so much bloody fanfare around this bit of footage that we'd waited to see, and then I had to sit there and then the camera cuts to me and I'm just like, it's a wallaby. Anyway. So I'm really sorry for that guy, but it was not a Tasmanian tiger. The other thing that Tasmanian tiger hunters love to do, there's a whole Facebook group of people that love to go out and hunt for thylacines, which is great. I love that people do that. I love that people think they see them all the time. Keeps the spirit of the Tassie tiger alive, and it gets it on the news. It makes people remember what an amazing animal this was. So I love all that stuff. What I don't love is that they go, I've got some shit. I saw a Tassie tiger and then I collected the shit that I found from that site and I'm going to send it to you. And I cannot tell you how many bags of shit that we've had to process. And I honestly, 5 years of my life, I reckon every day I'd come in, check my mail pigeonhole, and it just had new bags of shit in there. And they really look a lot like dogs, right? So it was dog shit 90% of the time. If we were lucky, it was a Tassie devil shit or something else. But we tested it, and now I have people in my lab now who just refuse to let me test any more shit. Actually, a box came in a couple of weeks ago and somebody really wanted this tested, and I said, could you please just test this one more box of shit? And the postdoc was going, I really don't want to do this again. It's just going to be dog shit. And I'm like, please. So anyway, so we did it. I was away at a conference and he texted me and he's like, not thylacine shit, guess what it is? And I went, please let it be human, because that would just be... I would have been, my hat's off to you, sir, whoever said that. That's pretty funny. But no, it was wallaby shit. So anyway, I do think that's no, they're not still out there. There's never been any scientific evidence of the existence of a thylacine at all in Tasmania since that last one extinct. And please don't send me shit. So if you think you see one, I love that for you. That's really great. I'm not testing the shit. We're done with that. It's not out there. The other thing I get a lot is like, oh, de-extinction. I've heard a lot about this before. I've actually seen a documentary about this, and it didn't go very well. So, as we like to call it, though, Jurassic Parkville. But I love nerds. Nobody laughs when I do that joke normally. So thank you. Thank you so much. I really appreciate that. But obviously there's a big difference between, bringing back a dinosaur that's been gone for 60 million years and bringing back the thylacine that's been gone for 88 years. So there's a stark difference between those two movies. If we could bring back dinosaurs, though, they did used to roam North America. So there could potentially be some good points to rewilding them at this point. I think, they might have some positive benefits back on that landscape. But I do get a lot of quotes from Jurassic Park. Nature finds a way. I love that one. I wish it did, because it's really hard work de-extincting science, doing that stuff. But anyway, I can just assure you that, DNA does have a half-life of 500 years. So if you do the calculations from a pretty big genome by 7 million years, no DNA left in anything anymore. Dinosaurs went extinct 60 million years ago. So it's a bit of bullshit about the old mosquito with amber. I hate to tell you. I actually really got an email last week from somebody who found a giant block of amber with tons of insects in it and said, "Do I want to have access to it?" But I'm like, there's nothing in it. it's all gone. So no dinosaur DNA in insects in amber. No T-Rex coming back anytime soon, and they're not going to eat our children. So I think, we're probably okay from that front. The other thing people say to me a lot is that you're playing God by doing this, which I think is a weird comment to make. I think, we certainly played God when we exterminated the thylacine and wiped them out. I think as humans, it's what we do all the time. Probably every single thing we do in science is playing God at some degree. So it's a bit of a weird argument to have that one. But I do think we're playing the part of good God, at least. We're definitely New Testament God in this scenario. We are not angry, spiteful, Old Testament, exterminate everything. We're coming in late in the picture going, I think we can try and fix some of these wrongs. So, I think it's interesting. And then the other thing that people say is, this is very expensive science, and it's super expensive work to do, and that we have a ton of animals that are on the brink of extinction, and we should be spending the money on the animals that haven't gone extinct yet and saving those, rather than putting that money into bringing species back that have already gone extinct. So the big point I want to make, and I hope you all take away tonight, is those two things are not at odds. So the de-extinction science that we're doing is saving animals on the brink of extinction right now. And these are all new technologies that we absolutely need to develop for marsupials, for our planet in general. So hopefully when I go through the rest of this, you see that there's a lot of work that we're doing, and it is all conservation science. And we get to bring back an animal that went extinct, which is great because that's another conservation move. But those animals, especially the thylacine, played such a key role in that environment that it can really help conserve all of the species in Tasmania in a way as humans that we could never manage ourselves. So it's really, really important science to do. So, de-extinction, no longer science fiction, no longer Jurassic Park, but thank God for that documentary because it really paved the way for a lot of the work that we're doing. But it is something that you can actually do. What you need to do de-extinction science is you've got to have a genome sequence for the species that's gone extinct. So you've got to have, it can't be gone for more than 7 million years. You can't go back too far. You need to have a close living relative of your animal that went extinct. And then you also need to be able to do assisted reproductive technology. So things like cloning, IVF, transferring nuclei, those sorts of things are things that you need to be able to do your species. And you can see there was a really nice feature in National Geographic, and they put the little thylacine just down here, which was awesome. Love that. So for those of you who remember the documentary, there was Mr. DNA who comes along and talks about how this thing works, and he actually was not that wrong, actually. It was pretty good. You would never fill a dinosaur, though, with amphibian DNA. I'm on, what the hell? I know here, and this is my room full of kindred spirits, that you would know this, that it would be bird DNA that you would fill it with, not ever amphibian DNA. But anyway, aside from that, Mr. DNA had a pretty good handle on what would happen. So what you have to do is you have to sequence the DNA of your extinct species. For us, that's the Tassie tiger. So we announced about a few weeks ago, actually, that we have almost completed the entire thylacine genome. We have just 45 gaps in its 3 gigabase genome, which is amazing. It's better than most living animal genomes. Yeah, it was a very big thank you. It was a very big feat. Huge amount of work. So we have an amazing genome, like incredible genome for this animal now. And then you use that genome to look around in nature and go, what is the closest living relative? And for us, it's this little guy up here called a fat-tailed dullard. I have a little video of it just to show you. It is a ferocious predator as well. It may be small, but it is mighty. So they're about 99% identical at the genome level to your thylacine. So you just align those two genomes, you figure out everywhere that those differences are, and then, that's what defines your thylacine. That's what made your thylacine so specific. We can't still create life where there is none, so we can't reanimate our thylacine, unfortunately, because that would be a lot easier. So what you do is you have living cells from your close living relative, from the dullard, and we just make that 1% of edits. I say we just make that 1% of 3 billion base pairs is a shit ton of edit still. We've looked, lots of those fall into regions of the genome that are repeat sequences and things that we know don't do a lot functionally. So we know there's about 500,000 edits that we actually have to make to turn this little guy into this big guy down here. And there he is. I'll put him into scale, just down there so that you can see. And so once you've done that, once you've made that 1% of edits, you've essentially got a thylacine nucleus, and then you can use things like somatic cell nuclear transfer, what we use to make Dolly the sheep, and go through a standing cloning protocol to then create a whole living animal from that cell. And that should be a thylacine. That is, 100% your thylacine genome at that point. So we're working on all of these bits down here and all of these bits up here to try and get this project to fruition. So at the moment, sequenced the whole thylacine genome, as we said before. We've sequenced the whole dullard genome. We have a colony of dullards thriving down in Parkville, and we've made stem cells from them. Stem cells is really good. First marsupial stem cells, which was really a great breakthrough for us. And we're now in the process of actually making all of those gene edits. And we're like 50,000 edits in of 500,000, but we're getting there. So it's a really big job, and this is probably going to take 10 or so years with the current technology to build a thylacine. I do think that technology is improving at a great rate, so it might happen quicker than that, but around about 10 years we should have a full thylacine nucleus. And in the meantime, we're working on all of the other assisted reproductive technology. So once we've got that, we're ready to go to turn that cell back into a whole living animal. So there's the fat-tailed, the very cute little fat-tailed dullards. So this is our surrogate cell, as I said, but it's also the surrogate mum for our thylacine. So some of you might be thinking, hold up, Andrew, you can see a problem here. But this is the miracle of marsupials, right? That this is a baby dullard on the day of birth. And they are, and thylacine, very closely related marsupial. That whole group of marsupials called dasyurids, they all have babies this size, and they're about the size of a grain of rice. And not your basmati jasmine rice, we're talking arborio rice, that short grain that I have down here. This is an arborio rice grain sized little baby that has to be born, crawl into mum's pouch. Just absolutely unbelievable. But what that means is that even that tiny, tiny little mouse-sized dullard can actually give birth to a baby thylacine, which is absolutely amazing. And then we scanned from museum collections all over the world every single little baby thylacine that we could find, and here's some of them here. And these are amazing things we can do now with micro CT scanning. We can have a look at everything, how they grew, how they developed, and we know it's going to outgrow mum's pouch in about 30 days, and then you'd have to hand rear that little baby. But we actually have milk that we use to hand rear marsupials. There are lots of orphan marsupial babies that we find all the time where mum gets hit on the road. And so we actually can buy powdered milk to raise those little babies. So that bit shouldn't be too hard. And one of the other things we learned is they actually had really enormous brains. Marsupials are pretty stupid, as it turns out. They have much smaller brains than placental mammals. And that's why we don't see that there are any domesticated marsupials or any marsupials that really make good human bonds. But the thylacine was very, very different. It has a very big brain. It's pretty common for apex predators because they've got to make a lot of decisions about hunting and pulling down animals and things like that. But it suggests that they could have actually been really, really good companion animals. And there's there's lots of First Nation stories about that and pictures of them with First Nations people. And so it would be amazing if we're able to bring these guys back and it turns out they're actually awesome pets or something like that as well. Much better than having a dog. So all of the work we're doing with the de-extinction work has these immediate conservation benefits. So as I was saying before, this is not just about bringing the Tassie tiger back, but it's about developing all of these new technologies that we can use right now to help our fragile marsupials that are all on the brink of extinction. So we're developing a lot of the assisted reproductive technologies, or ART, and we're using the Done Art to do this. So this is our Done Art project. Come on, that gets a really big laugh from the reproduction crowd. I was really counting on you for a laugh there. But so we're doing a lot of the work on just trying to get embryos to grow and all those sorts of things that will have major implications for our ability to captive raise marsupials and re-release them after things like bushfires and floods and other things where they've been removed from the landscape. The other thing that we've been able to do, as I mentioned before, is make stem cells for the first time in marsupials, really bona fide, proper stem cells for marsupials. And as a result of that, we were able to get a grant to set up, an ARC linkage grant to set up a big biobank, Australia's first living cell biobank, where we're working now with all sorts of different conservation groups to go out when they're doing population surveys of marsupials, but any animal in Australia. They can collect cells from an ear punch or a foot scrape. They can drop it into a little tube, put it in a post pack, put it in the post office, and it will get delivered to Museums Victoria. We have some technicians there that will then grow the cells up for us, freeze those down, and we will keep those living cells forever. And that means that that species hopefully can never go extinct. We've always got living cells for it, and that we never have to do this grueling, bloody de-extinction project of re-engineering something back again, because we've actually got living cells frozen down from those species. So this has been a fantastic immediate outcome that we've been able to have from the work that we've done. The other work that we've done, and this is actually championed by Stephen Frankenberg, who's sitting right at the very back of this auditorium here today, is using that DNA editing technology that we can do now so easily in marsupials to help save our marsupials from some of the terrible things that they have to face in the Australian environment. So a great example of this is the northern quoll, this beautiful marsupial that lives in the northern parts of Australia, and they are one of the animals that's predicted to go extinct in the next decade because they're eating cane toads. We hate cane toads. Introduced pest, horrific species, a toxic toad, and none of our species have any resistance to this introduced group of toads that have been just placed in Australia. But where these toads naturally occur, and there are tons of mammals that live alongside them, all of those animals over hundreds of thousands of years have evolved resistance to that cane toad toxin. And if you have a look in the genome, there's really one key receptor. There's a couple of genes, but really one key receptor, this ATP101 gene, just one gene that confers you either being completely susceptible to cane toad toxin and dying if you eat them, or resistant to that toxin. And in fact, there's only one amino acid, one residue in that protein that either makes you toxin resistant or sensitive to that toxin. And so what we did is we proposed to engineer that resistance then into marsupials. You can actually do this in cell lines. Cells will die if you put the toxin on top of them, but also if you can engineer resistance, the cells can survive. And so in this graph here, we've got just our normal, this is our fat-tailed dunnart, so we engineered our fat-tailed dunnart to be cane toad toxin resistant. You can see in this guy here, if we have just its normal cells, we expose it to cane toad toxin, the cells eventually die. At this point, this is where our cells are all dying in the flask. We can make some edits that occur in a few different lizards and reptilian species, and we can make them about 12 times more resistant, so about 20 times actually more resistant to what they are if they don't have those edits. And then we can make this single base pair edit in their genome. This just changes one single amino acid, and this is what most mammals have in their genome. And if we do that to a fat-tailed dunnart, we can make that completely resistant, those cells completely resistant to cane toads from just one single base pair edit. And so we're now doing this in quolls. We have quoll stem cells that we've made. We're making that edit in the quoll stem cells, and we'll be able to demonstrate it will have the exact same effect in those guys as well. So this will be a really important example of where we can use genetic engineering thoughtfully to engineer our wildlife to survive this peril. The truth is, if we do nothing, we will lose the northern quoll. We can do some genetic engineering. We can change just one base pair, and that one base pair that we're changing would naturally evolve. They would evolve that resistance if we had another 100,000 years before that species went extinct. Unfortunately, we don't. There's so many bloody cane toads that it will kill them. They'll be wiped out before they have a chance to go and evolve that particular resistance. And so we can make that guy resistant doing it. And this will be a great example of genetic engineering to save our wildlife. It'll be an ambassador for doing genetic engineering in marsupials. But also, not only will it save the coal, but they can actually eat bloody cane toads, which will be absolutely fantastic. Hopefully, these animals then can help stop the spread of these terrible species across our planet. So it'll be a really great outcome. So that's it. I just wanted to say de-extinction science is a really important new area that I hope that everybody here is willing to be open-minded about, where it can really contribute. And we are creating these new methods that are helping us conserve the species that we have got left, preserving the animals that we have and their biodiversity, and also restoring some species, those really important species that went extinct that are really important in certain ecosystems. And then the truth is, with everything that we've done in the world, we really need to consider these other tools that we have in our toolkit if we really want to preserve things on our planet. And I thought could potentially lead to the cure for IBS, but probably. And that's it for me. Thank you very much.
What an amazing talk. Keep that clock going for anything. All right. Does anyone have any questions for Andy? Ooh, lots of hands.
I love it.
You first.
I was just wondering if we actually have the full sequence right, or is it more important to get the genes responsible for growth and growth? Second, you get something that's the right shape and everything else that does that.
Yeah.
So do we have to get every single amino acid correct in your sequence?
No, and I love you for asking that question. I do think for putting an animal back into the wild, we have to make all the edits. But I think we'll make a thylacine, something that really looks like a thylacine, because we know the changes that really probably do those big, change the morphology, change the size, change the shape of the skull. In fact, we have a lot of people who are working on exactly that, like figuring out what all those genes are. But I think if you want to put that animal back into the landscape, you've got to make all of those changes, because even the ones that occur in repeats and bits of the genome that we all think are dull, the more we learn about the genome, the more we learn that some of those things do actually have important functions, things that we don't understand yet. And there are other things like brain function. We don't, you can make a change, but we don't know what that looks like. It's probably a change in, chemicals that are being expressed in certain neurons in the brain. So we will have to make all of the changes, but I think we'll have a thylacine, really cool looking animal, before we have the one that actually goes back out to the bush. Yeah.
Oh, very cool. We've got some questions at the back. Maybe I'll go to the back. Yep.
About the behaviours of the reincarnated thylacine once it is reintroduced. So there's obviously within the genetics, final stuff like being able to walk and swim or whatever.
Yeah.
And there's also the known behaviours. So what are the considerations that you need to have on the reasons of having those learned behaviours and
Yeah, so what kind of learned behaviours might you have to consider before you just chuck a Tassie tiger out in the wild?
So marsupials are pretty dumb, which is a good thing in this case, except thylacines have big brains. So I'm like, damn it, would have been good if they had small brains. But things like the really important behaviours for an animal, like the hunting behaviours, breeding behaviours, those things are super hardwired in our brain. So all of those really, really fundamental ones, they're not learned. They're all really, truly hardwired. And, there's tons of examples of this, but, we have lots of rewild lions and tigers and things that people hand raise and put back into the environment, and they are able to, readapt to the natural environment and thrive once again. A great example of this is house cats, though. Like many of us here might have pet cats, although we're a bunch of nerds, so maybe we all hate cats. Oh, boo! Cats in the house, fine. Cats outside, no.
Oh, yeah.
All right, yeah. But cats are a great example of something that's been fed from a tin for, hundreds of generations now. But if you wiggle something in front of them, they're still a pounce predator, right? That shit is hardwired in their brain, no matter, how captively bred these animals have been. So those things, hunting behaviour, mating behaviour, the really critical ones, They are hardwired, and we would never release that first batch of thylacines, back into the wild. So by the time we have a population of thylacines that you would think about rewilding, they should have had a few generations of being raised by other thylacines that hopefully they'll pick up some of those behaviours and things again. But we have no good records of what their behaviours were like. So I can't really get it wrong. Great.
We love that. We love that. All right, here.
Great talk and great work, obviously, but 10 years is annoyingly long.
I'm sorry.
No.
Just a comment.
How are you doing in the editing? Is it literally like CRISPR by CRISPR, base by base? So what are the new technologies that's going to speed that up? Do you think synthesis? I know the yeast guys have a fully synthetic.
Yeah.
Ooh, how can we speed up this process?
If I told you, I'd have to kill you, Sam. But no, the group I work with, Colossal Biosciences, which is a huge big biotech company in the United States, founded by George Church, who's an absolute genetics superstar. It was one of the fundamental inventors of CRISPR technology. They have a lot of tools that are really accelerating this process, where you can do editing en masse. So you can multiplex, and make 100, 200, 300 edits at once. That's still not enough, though. Like, if you do the maths, we still need to do bigger scale editing. Artificial DNA is potentially going to be a game changer. So there's a few companies out there who think they can synthesize megabase long fragments now. I don't think it's going to be that long before you could potentially synthesize an entire chromosome, for example. And then that's a game changer, right? Then you don't have to do any of the bloody editing. You just make your artificial constructs and, piece together your genome, basically. So that would be absolutely phenomenal to do. So, yeah, there's a lot of things. I am fairly confident that in the next decade this will get quicker. So hopefully it won't take that long.
It definitely sounds like something out of a sci-fi movie. Yeah. We might go up here. Yep.
Thank you so much for the talk. I want to know about the gene editing techniques that increase resistance to environmental toxins. Do we expect those edits to carry on into the offspring?
Yeah, so one of the great things about these...
It's totally fine. The question was...
with the gene edits to cause toxin resistance to the cave toads, is that going to get passed on to offspring? So, yeah, one of the things that's so good about this is because it would be so highly selected for. Basically, if you don't have that particular allele in one of those genes, you're going to die at some point. You will eat a cane toad and die. And so almost every animal in the landscape you'll have to create a lot of quolls that have this and release them, multiple times, but it would spread very quickly through the population. It would be such a huge advantage for animals that had that resistance that this is something that would actually spread very quickly. You wouldn't have to think about doing a gene drive or any other method to make it spread more quickly. It will actually be super selected for.
Amazing. Maybe we'll just have one more. Someone with a burning question. Who's got the biggest hand? Oh, we'll go with you because you kind of backed out of it. And I want to know your question.
Thank you. Great talk. I guess you're creating a Tasmanian tiger from a cat tail dunnet, and someone done the whole process as a proof of concept from start to finish with two more closely related things like two species of dog or even like a brother and sister animal.
Yeah,
Ooh.
That's a really good question. So the question is,
you get too eager.
The question is, like, these are quite far apart, like thylacine and thylacine, really, with that 1%. Like, it actually, in evolutionary terms, there are species that are much more closely related. Have people done this with more closely related species? No, they haven't. And, I actually suggested, and Stephen Frank makes that, we suggested maybe we bring back one of the extinct rodents first because, we're so good at genetic editing in mice. They're very closely related. Nobody wants that shit. They all want a virus, right? So we tried to do that, but everybody's like, no. No. People are working on the mammoth. So the woolly mammoth, the fun fact you can use at your next dinner party, is the woolly mammoth is more closely related to an Asian elephant than Asian elephants are to African elephants. They are very closely related together. So it's not going to take the guys very long at Colossal to make a mammoth cell. They're like 3/4 of the way there. But then turning that cell back into a mammoth, it's a 22-month gestation, a 500-kilo baby. That shit's hard. That's where you want your arboreal rice grain, right? Like that. It's going to take us a long time to edit, but the next bit, we're like, we got that bit. The mammoth, that is hard. And then, there's a lot of incompatibility. It has a giant placenta. Marsupials don't even recognise that they're pregnant. They just have babies pop out at one point. So there's a lot of advantages to using a marsupial. But yeah, it would be, you're exactly right. That is a brilliant question, but nobody wants to do it.
Can I ask the very, very, very last question? Being a scientist, I know how tough funding is to get. Where did your funding come from, and how much does this cost?
Okay, that's a fairly long story. Do I have a few minutes?
1 minute.
Okay. So I was at home during COVID lockdown, and I had a 7-year-old at the time, homeschooling. My life was absolutely terrible. Like, I was teaching first year biology. We had 3,000 students in first year biology at Melbourne, and I was the coordinator that year. It was like the worst year of my life. And I got an email from somebody saying, hey, I saw on on YouTube that you do some stuff with thylacine. I'd love to have a chat to you. And I was like, normally I would be like, delete, but I was feeling lonely and craving grown-up attention. So I truly, I would never respond. I get so many insane emails. And I responded back and I was like, sure. So he's like, let's set up a Zoom. So I join, he joins, he has no name. It's like Hot Girl 69. I'm not even kidding. Like, I'm just looking at it going like, anyway, he asked me a whole bunch of questions about de-extinction and what I'm doing and what I'm working on. Went for about half an hour. Again, lonely. So I was like, oh, I'll do it, bit of human contact. And then he went and that was it. And I was telling everybody in the lab and they're like, this sounds really sketchy. And I'm like, it was sketchy and didn't tell me who his name was or something. So then I get an email about a week later and he's like, can we have another chat and can I bring my brother this time? And I was like, I'm not into that. Like, this is weird. So anyway, I'm getting lonely. So I'm like, new experience. I'm at home in COVID. So we jumped on, had another chat. His brother was there. They both asked a lot of questions. He had no name, Hot Girl, I don't know, 27. And it was just bizarre. And then that happened. And then he said, could I put together a plan for how much it would cost to do this? And I'm going, I don't know, like, who is this person? They don't tell me anything. So I've spent honestly like 2 hours and put together like a 3-page booklet thing and just sent it over to him saying this is how much a postdoc costs, this is how much the science costs, we need 10 years worth of funding to do this and put it all together. And then he said, can we have another call? And I'm like, yep, sure. So, at this point, I've lost all dignity, so I may as well jump back on again. I've done it with his brother, like the whole thing. So then we jump back on again and he's like, I would love to fund you. I want to give you $5 million. And he's like, I'm truly the luckiest scientist in Australia, like, honest to God. Like, this does not happen.
Yeah, when can I get myself one of those?
He's like, do you want it in 5 years or 10 years? And I was like, for over 10 years. So we've got $500,000 a year for 10 years of funding. But then he went, right, he's like, do you have any questions for me? And I'm like, yes, who are you? So then he was laughing, told me all about his backstory. He runs CoinSpot, very huge in Bitcoin, made a ton of money and wants to give back, loves awesome science, big nerd, love him. And then he hung up and then I was like, what do I do now? So then I called the philanthropy people at Melbourne University and I'm like, I think I've just got a philanthropic donation. And they're like, okay, how much? And I'm like, $5 million. And they're like, who is it? And I'm like, his name's
Hot Girl 69.
Russ, I think. And then they're like, when did you meet with him? And I could see them going through it. And then I started going, did I imagine this? Am I so depressed and lonely at home that I've made this whole shit up?
Your daddy or?
All of this. It was real. Everything went great. And then I announced that I was doing it. We had a big announcement. It went absolutely like crazy over the world. It was this huge viral thing that we're doing it. And then this big company in America, Colossal Biosciences, were like, oh shit, we're working on that. We need to talk to you. So I went and met with them and then they said, we need to be the first company that does this, so we're just going to fund you more because we just want to be a part of that. And so it's just been amazing. They're the nicest guys as well. I love it. I get to travel all over the world. We do incredible science. We're going to do the mammoth, the dodo, this guy, and it's going to be awesome. And I'm sorry if they end up killing everybody.